Thank you everyone for joining us at our webinar today covering a faster workflow and improved coverage for copy number analysis. My name is Sean Case and I'm the product manager for our CytoScan portfolio here at Thermo Fisher. And I'm joined by my esteemed colleague, Debbie Black, who was a member of the R&D team who developed the CytoScan HD Accel product, which we will go into more detail later in the presentation. So as an agenda, I'm going to give a quick introduction and overview, and then I'm going to hand it off to Debbie to go into a lot more technical details on the workflow, performance, the expanded reference model that we built, as well as some actual examples pulled from Chaz, and then we'll wrap it up with a summary and Q&A. Now, for existing CytoScan customers, this is nothing new, but for anyone that may be new to CytoScan or the applications in general, I just wanted to clarify the applications where the CytoScan products are used in. Today, we're specifically talking about Accel, and those three applications are prenatal, postnatal, and oncology research applications. And in the latter, in oncology, that primarily tends to be hematological oncology. So here at Thermo Fisher Scientific, we feel strongly that microarrays aren't going anywhere anytime soon. And we showcase that with our continued investment and commitment to innovation. More recently, with a launch at the end of last year, was our CytoScan Air product, which stands for Automated Interpretation and Reporting. And while Chaz is a fantastic analysis tool, it doesn't bridge the gap between the interpretation classification piece. One of my colleagues had a great analogy of simplifying what the Air solution brings to the table. And it essentially breaks down to without Air, when you're manually going about it, it's like being in a library, having to go around, pull books one by one, go through articles, pull out all the relevant information and curate that all yourself, which is very time consuming. Whereas with Air, it's like someone handing you a folder with all of that information already curated and organized, making your job that much easier. The latest innovation and launch is our CytoScan HD Accel product, which just launched last month. I won't speak too much more about it as we're going to go into much more detail throughout this presentation. And then next coming down the queue, which will launch at the end of this month, is our Chaz 4.5 product, which is the data analysis software used for the cytogenetics suite. So if you haven't picked up on it already, the theme and primary objectives behind these innovations and launches are to really streamline and improve the efficiency and productivity from operational standpoint within your guys' laboratories. So what Chaz 4.5 is going to provide is the automated enablement of the transfer of data files from the scanner itself to Chaz. And then for those users that have the Air solution enabled, they'll also be able to set it up so that the data files automatically transfer to the Air solution as well. As of today, the files sit on the scanner, and then when the end user comes into the lab or office, they have to manually transfer those over, which takes anywhere from 6 to 10 minutes, depending on the array type that was used. And calculating your own volumes within your labs, you can easily determine the amount of time savings that this will offer. The objective behind this slide is just to really provide insights that we're not just providing a kit. We're providing a holistic solution that has everything that you need to go from DNA to results, starting with a modular instrument, which we'll go into more detail on the next slide, but also includes a continuum of solutions and different array types that all can be ran on the same instrument depending on what your needs are, along with the downstream data analysis piece called Chaz, which stands for Chromosomal Analysis Suite. And with that, I'll hand it over to my colleague to review the workflow and to dive into more technical details. Thanks, Debbie.
Thank you, Sean. So first, I'd like to get into more details regarding the workflow, starting with highlighting the instrumentation which is being used, providing flexible, scalable, and easy to use instruments, starting with sample collection and the genomic DNA extraction. There's no prescribed method. The customer is enabled to use their own favorite workflow as long as the genomic DNA is relatively intact and free of PCR inhibitors. The purified genomic DNA is then used for target preparation, which we'll go into in a little more detail in the coming slides. This is a manual step followed by array processing, which includes array hybridization and automated washing, staining, and scanning. And then followed by that, the cell files or data files are analyzed using CHAS as well as visualized in CHAS, providing a genome-wide view and analysis of chromosomal aberrations and SNP variants for copy number determination. And the GCS 3000 is very modular and scalable in the sense that the hybridization oven can support up to the processing of 64 arrays at one time, and the GCS 450 Fluidics can accommodate 4 arrays for washing and staining, but you can configure up to 8 fluidic stations and run them simultaneously to support the processing of 32 arrays, followed by placing these arrays into the scanner with the autoloader to support the scanning of 48 arrays in a single run. And so this instrumentation, the GCS 3000 platform, is supported by our gene chip data collection software, or GCDC, which was recently launched last year and enables walk-away array processing with remote monitoring capabilities. Now to go into a little more detail regarding the HD Accel assay and array, I'll just present the highlights here, and then in the coming slides provide additional details. So the HD Accel assay allows a 2-day turnaround time. It incorporates a plate-based purification method, which reduces pipetting steps and mitigates the risk from sample swapping. It uses 100 nanograms of genomic DNA, which is about 50% less than comparable chromosomal microarrays on the market, including CytoScan HD, and it leverages an updated reference model file, which contains a diverse set of sample types, and will highlight the benefits of this later in the presentation. In addition, the CytoScan HD Accel array was newly designed with increased probe coverage to allow for more accurate copy number breakpoint detection, and provides coverage in over 5,000 relevant regions as classified by professional databases such as ClinGen and Decipher. This just goes into more detail now on the workflow. On day 1, we have target preparation, and after that, we take that target and we hybridize overnight to the array and wash and stain and scan on day 2, with the data files being available for analysis on day 2. So the DNA target is ready for hybridization in under 8 hours. And this assay provides some schedule flexibility as well, as there are potential stopping points in the assay that allows the operator to freeze the plate for up to 10 days if a 2-day turnaround time is not needed. In addition, the user guide provides workflows supporting the processing of 8 to 24 samples, although fewer samples could be processed at one time. And so we have a workflow that provides schedule flexibility with convenient stopping points. Now to go into a little bit more detail on the target preparation step, we share on this slide the various steps, which start with digesting the genomic DNA with a restriction enzyme, ligating adapters to this fragmented DNA to allow for targeted PCR for complexity reduction, generating PCR products of 150 to 2,000 bases, and then going on to purify the PCR product. We then quantitate as a QC check, and then fragment it into sizes that enable efficient hybridization, fragments of 25 to 125 base pairs, followed by fluorescently end labeling these fragments to support hybridization on the array. There are some key target preparation improvements that were incorporated into Accel. We have a new assay reagent kit that has improved biochemistry to support shortened assay incubation times. In addition, the PCR product is purified using a magnetic plate-based method as compared to a tube-based purification method. And this has several advantages. One, it minimizes the potential for sample swapping, and it also involves fewer pipetting steps, reducing pipetting at this step by about 75% as compared to tube purification. And this contributes to the assay time savings. In addition, the recommended magnetic plate is compatible with liquid handlers for those customers wishing to develop their own automated target preparation solution. So let's now take a look at the CytoScan HD array and the extracted information provided by Chaz. The CytoScan HD Accel array provides genome-wide comprehensive coverage using a high density array that contains approximately 2.8 million markers, including 750,000 SNP markers and 2 million non-polymorphic markers, which includes that 100,000 new probes that were added to the Accel design as compared to CytoScan HD. This is referred to as a hybrid dual probe design, which includes, having the copy number probes as well as the SNP probes for based on the SNP probes being selected based on their high minor allele frequency to enable good separation of allele tracks. And this design provides for confident breakpoint determinations. In addition to providing independent allelic or SNP confirmation of copy number changes, and also supporting the high resolution detection of the loss or absence of heterozygosity or LOH, AOH analysis, as well as enhanced detection of low-level mosaics, as well as genomic contamination. And we'll highlight some of these applications in the coming slides when we're looking at those Chaz use cases. So Accel was designed to provide the same robust performance as CytoScan HD, although it incorporates the design using the most current human genome build, HG38, as well as supporting the use of a lower amount of input DNA, 100 nanograms. And so our minimum resolution on this array is a 25 kb for losses and 50 kb for gains. We also have good resolution for runs of homozygosity, as well as the ability to detect low-level mosaicism. The Accel array is supported by Chaz 4.4, and we have the log2 ratio track, which we can use for looking at copy number, as well as the allele difference track and B allele frequency track to allow the SNP contribution in terms of interpreting the data. The new design of the CytoScan HD Accel array, based on the current genome build and with the addition of the 100,000 probes, results in a comprehensive chromosomal microarray that is providing up-to-date coverage and includes 100% coverage in the ClinGen and Decipher databases and coverage in over 5,000 relevant genes. And the design of this cell array, we were responding to customer input, and we were adding additional probe coverage to genes of interest. And these are some of those genes listed on the slide. Several of these genes are transcription factors, such as SOX3, which is associated with autism, and the zinc family member 2, or ZIG2, which is associated with cancer. I'd like to discuss another highlight of CytoScan Accel, which is its diverse reference file, and how this diverse reference file supports the generation of high quality data for the multiple sample types used by our customers. So what is meant by a reference? A reference model file contains data representing phenotypically normal individuals, and it's used to establish the signal baseline for the normal diploid copy number state of 2. And in addition, the reference is used to establish the SNP genotyping clusters. And what we want in a reference is diversity. We want to capture normal typical sources of variation, and in addition to biological diversity. And this provides an overview in the slide of how diverse the CytoScan HD Accel reference is, with 656 samples captured in the reference, about twice the size as the CytoScan HD reference. And this reference includes not only blood and cell line samples, but additional prenatal and postnatal samples. Sample types of amniotic fluid, buccal, CVS, POC, and saliva. And we included the diversity related to operators as well as reagent lots and instrument diversity. And on this next slide, we will highlight how the CytoScan HD Accel reference not only captures that sample diversity, but also ethnic diversity. And so before I go into the slide, I need to say a little bit about principal component analysis, or PCA. So principal component analysis is a statistical multivariate analysis that reduces large complex data sets into 2 dimensions and enables visualization of the data so that we can see correlations between the data set and identify any potential sources of variability. And so what we're looking at in this slide is we're looking at a PCA plot, looking at the genotyping information. And on the left is representing the CytoScan HD reference samples. And we can see in PCA plot 3 distinct clusters, which represent the populations from the HapMap samples used to create this reference. We see tight clusters for Asian, Caucasian, and African populations. And now when we look at the CytoScan HD Accel reference data projected into the HD genotype PCA space, we still are able to detect these 3 clusters representing these populations, but in addition, we see these additional data points between these pure populations, and this represents admixture individuals. And so the CytoScan HD Accel reference not only captures the ethnic diversity present in CytoScan HD reference, but it actually provides additional ethnic diversity. And we'd like to share the steps involved in building a reference, and with the goal that we want to capture the potential sources of normal variation in the reference and then correct for this variation. And so building a reference is an iterative process. We first examine the data, make sure it's of high quality, and perform PCA analysis of that reference plate data, including plates that have been excluded from the reference, and we compare this against current intermediate reference. We then go on to identify normal samples that meet a reference independent QC metric to ensure we're putting high quality data into the reference. And we include these data files meeting this criteria in the next version of the reference. We then reprocess all the existing data using this newly generated reference file, and we re-examine the QC and PC analysis of this reference plate against the new reference. And we then generate another batch of data, and we repeat this process. And we repeat this process until all normal variation appears to have been captured and corrected with data normalization algorithms. And during the reference build iterations, we can see from this PCA analysis, looking at the signal log2 ratio data, that most sample types appear to behave similarly and co-cluster in the PCA plot. And so what we're looking at here are the various sample types color-coded and overlaid with the entire sample set shown in gray. And so we can, for the most part, see very good co-clustering of these different sample types amongst each other, except for saliva. What we visualized early in the reference build process is the saliva sample types appear to show outlier behavior or a batch effect. However, if we go on and further iterate versions of the reference and continue to capture the variation associated with the sample saliva sample types, this variation could be normalized. And what we're showing now is the data for the final released version of the reference file, where we can see that we now have all the different sample types co-clustering together and minimal variation observed between the sample types. And this final reference is minimizing batch effects due to sample types and results in having a robust reference that can support high quality assay performance. And so high quality data is first evaluated using array QC metrics. And the CytoScan HD Accel leverages the QC metrics that we use for CytoScan HD, and they relate to copy number performance, MAPD, and waviness SD, look at log2 signals, and evaluate signal noise in the data, either short range or long-term signal noise, represented by MAPD, waviness SD. And SNP-QC is a measure of allele genotype quality. So lower MAPD and waviness SD values are desirable, and higher SNP-QC values are better. And this is a slide demonstrating how how the QC metrics improve with the reference file iteration. We are showing MAPD, one of the array QC metrics, and we're looking at 6 different sample plates analyzed with 4 reference iterations, just labeled as A, B, C, D for simplicity. And what we can see is that overall, the MAPD values improve or become lower with each subsequent reference iteration. And this is very easily depicted in plate 6, where we can see that decrease in MAPD going from the reference iteration A to reference iteration D. And using our final commercially released reference, we see that all sample types are displaying high quality array QC data, which is predictive of good copy number performance. And this will be illustrated in CHAS in the coming slides. But first, I'd like to share, as Sean provided an overview of our CHAS or chromosome analysis suite software and some of the key features. It really provides a comprehensive analysis and visualization. It was designed for cytogeneticists by cytogeneticists for research. It allows that analysis of the cell files from the scanner and enables a variant classification, in addition to linking to external websites and databases. And as Sean mentioned, CHAS 4.5 will be coming available shortly at the end of this month and will support the automated loading of cell files into CHAS for hands-free analysis with connection to Franklin reporting. And so this will free up valuable time of the lab operators from data analysis and variant interpretation. So now I'd like to just share a few of these use cases using the CytoScan Accel. We'll first look at the detection of gains and losses on this array. And so this is a sample, it's a bone marrow sample illustrating the ability to detect a complex loss. We have a copy number 0 segment that's nested between 2 copy number 1 segments, and that's depicted in CHAS. On the first track, what we see are the copy number loss segments shown by these boxes, followed by the log2 ratio, with the log2 ratio of 0 representing the normal diploid copy number state of 2. And so what we can clearly see in this example is the deletion, copy number 1, followed by this small copy number 0 segment. In this slide, we're able to highlight, by increasing the probe coverage in CytoScan Accel, how we can now detect this small 150 kb loss with more accuracy due to more accurate breakpoint detection. And what we're seeing on the top track is CytoScan HD Accel data as compared to CytoScan HD array data in a lower track in pink. What's highlighted in the box is the additional probe coverage that we have on CytoScan HD Accel, and this additional probe coverage is allowing us to accurately call this chromosomal loss over this important gene region, whereas the lack of probe coverage in this area on CytoScan HD does not limits our accurate breakpoint detection. We also see another example of this where we're looking at a small 70 kb gain, and we're able to accurately determine the breakpoint for this gain in CytoScan Accel by having this additional probe coverage over the gene regions as compared to HD, which lacks probes in this area. We also, in our development studies, were interested in investigating maternal contamination and how we're able to detect this on the CytoScan HD Accel. So maternal contamination is problematic, especially for prenatal samples such as amniocentesis samples or chorionic villus sampling due to the presence of maternal cells. Because this contamination with maternal cells can make it more difficult to detect chromosomal aberrations present in the fetus. So we wanted to investigate this further by performing a simulated maternal contamination study. The purpose of this study was to determine the approximate upper limit of maternal contamination that can be tolerated while still detecting a known chromosomal aberration in a child. We wanted to also determine the approximate upper limit of maternal contamination in a sample that can still pass our SNP-QC array metric. And so the study design involved using 2 cell line samples, a mother that was normal diploid, and a child that was a trisomy 21. And we created mixtures of the child mother and performed triplicates of these mixtures. And on the right, we can illustrate these different mixtures where we go from having 0% maternal contamination to up to 30% maternal contamination. And we first evaluate the QC metrics, looking at MAPD, waviness SD, and SNP-QC. And what we could see is, for the most part, MAPD and waviness SD are not highly impacted by the sample mixing, but as predicted, SNP-QC is. And we're able to see that beginning about 25% maternal contamination, we start seeing a failure of one of the triplicates, and complete failure of all 3 of the replicates with a 30% maternal contamination. So the simulated cell line mixing maternal contamination study using our 3 technical replicates, when examined in CHAZ, shows that we can visualize the trisomy even in the presence of 30% maternal contamination, despite the poor QC from SNP-QC that resulted from sample mixing. So mosaicism is another application of interest. Mosaicism involves the presence of 2 or more genetically distinct cell lineages within an individual derived from a single zygote. And this is illustrated on the right-hand panel. And we can see somatic variation observed at all genomic scales, from point mutations to aneuploidies. And it's challenging to detect gain or loss of a small copy number variant in mosaic samples, especially when present in only a small fraction of the total cell population. However, what we can see in this example on a bone marrow sample is that we are able to detect a low-level mosaic gain on chromosome 10. And we can see this from the smooth signal track down in the middle here, which indicates a copy number of 2.11, representing a 15% mosaic gain. We're also able to detect a copy neutral of loss of heterozygosity. And what we're seeing in this illustration is HD Accel data on top as compared to HD data on the bottom. And we get the same robust detection of LOH in HD Accel as we do in CytoScan HD. And in fact, in CHAS, we're able to report on the autosomal percent LOH, and we can see that we're getting the identical percentage of percent LOH in both HD and HD Accel. And so would like to summarize by highlighting the benefits of CytoScan HD Accel Suite. It provides robust and reliable, high-quality data using a reference model that's diverse, including blood, buccal, saliva, amnio, CVS, and POC, as well as cell line samples. It improves workflow efficiency and boosts productivity, leveraging a streamlined workflow that provides 2-day sample to insight. It is able to leverage using low amounts of genomic DNA input, about 50% less than other chromosomal microarrays on the market, and the use of a plate-based PCR purification method as compared to tube purification reduces the risk of sample swapping. And we have, with the CytoScan Accel array, updated comprehensive coverage that provides coverage in over 5,000 relevant genes and provides genome-wide copy number information with increased confidence in these copy number results. And so I'd like to thank you for your attention and take any questions that you may have.